Hi, everyone. Welcome to the Cerebellum seminars. Today we have as our guest Andrei Khilkevich. Hilkevich, I should say. Apologize. Andrei received his bachelor's in physics from the Belarusian State University, and then he came to the United States, where he did a PhD with Michael Mock at the University of Texas at Austin. While he was there, he made some major discoveries regarding learning of sequences in the cerebellum. He demonstrated that you don't need to have a error signal from the climbing fibers to learn a sequence that's made up of multiple elements, but that the movements itself can become the guides for learning of the second and future elements of the sequence. After he finished his PhD, he went to London, where he became a postdoc with Tom Mersick-Flogel at the Sainbury Wellcome Center at University College London. And over the course of 4 or 5 years, they made major discoveries regarding brain-wide activity that leads to decision making, and really changing the way we think about decision making as not being associated with a specific region, small region of the brain, but rather brain-wide activity throughout. So, Andrei, thank you so much for presenting your work.
Thank you, Reza, for the kind introduction and for having me here. It's a pleasure to show my work to all of you. And also, I know that by convention, the questions are reserved towards the very end, but because what I'll be showing is much less cerebellar focused as usual seminars are, I really don't mind if people have questions through the talk, especially if they're clarifying questions. All right. So with that in mind, so what we want to study in this project is decision making. And examples of decision making or deciding when to act are, for example, if you cross a somewhat less regulated traffic, or if you're in London and there's a bit of rain, but you're always evaluating how much rain is sufficient for you to open the umbrella. Right, these are the examples of the everyday tasks. And on the conceptual level, we can think about it as a transformation of the sensory input through some internal decision-making process. Sorry? Decision-making process into the action. And what we're interested in as an overarching goal is to understand how these processes map onto the distributed brain areas all over the brain. That's, of course, I'm not going to answer this question, but the hope is that the current work will provide some foundation and insights towards this question. Okay, so to walk you through the behavioral task that we use here. We have a head-fixed mice on a running wheel that observe a grating that drifts either upwards or downwards. So to give you an idea what they see, this is what they look at. And what they need to detect is when grating starts to go in faster, like this. As you notice, this is example of the single trial, right? So the speed, that's what's plotted here, the speed, the temporal frequency of the stimulus, it's noisy, and it's noisy intentionally. So there are fluctuations around the mean, and at some point, the mean speed changes, right? And so this mean increase in speed is what mice need to detect. They detect it by making a lick, just to say that, and they get rewards, which is the soy milk, if they lick correctly, right, during this time period. Now, we want to design the task such that the movement, the movements don't obstruct the neural recordings, right? And so what it means is that we want the mice to be very patient, such that they're on the running wheel, and they can move on a wheel, but if they do move during this trial, the trial restarts, right? So they cannot move if they want to get the reward. They also don't know when this change in the stimulus speed will happen, because It happens from the start of the trial anywhere from 3 to 15 seconds, which is a long time for a mouse to wait on the wheel, and yet they do that. On top of this noise in the stimulus speed makes very ambiguous for the mouse for whether the change has happened in the stimulus speed. And so the hope is that this will encourage them to integrate, right, over time, the stimulus to figure out whether this increase has happened or not. Okay, so behaviorally, this is some psychometric and chronometric curves. So this is fraction of hits as a function of change magnitude, for example. So as expected, the stronger the change, so this mean increase, right, in the stimulus speed, the easier is for mice to detect it, and then they react more faster. Even more importantly, if we look at the leaks that precede the change period, right? Because again, the change time varies trial to trial, so mice never really know when will it happen. And so these leaks that happen during this preceding period, we call them early leaks. And yet those leaks are not really random, because when we look at the average stimulus that precedes the leaks, we see this overall increase in stimulus speed, right? So they react to this randomly occurring increases in the stimulus speed. Basically what this shows us is that mice are constantly paying attention to the stimulus and trying to solve the task. And that's also why we don't call them false alarms. Okay, so I want to touch very briefly on the behavioral strategy, what the mice use, and then we'll jump to neural recordings. And so what this means is I'll outline kind of what we did in terms of the behavioral analysis, but I'm not going to go into details of the analysis itself. But if someone has the questions afterwards, I'm very much happy to go there. So what are the options of behavioral strategies? One option is that mice integrate the stimulus over some temporal window, right? Another option is that they simply use outliers, right? So for example, this, what we call, okay, one thing I haven't said to you is what we call the pulses. So the stimulus speed is consists of this 50 millisecond long bits of temporal frequency. So every 50 milliseconds, the stimulus speed changes, right? So in each of these instances, we call the pulse of temporal frequency. And so mice can simply detect, for example, some fastest pulses over some threshold, right? So that's a possible behavioral strategy. And so Ryan constructed this, what we call the outlier detection model, which basically poses that there is some certain threshold over which, if pulse crosses the threshold, mice make a decision that, yep, the event has happened, I should make a lick. And then there is some variable motor delay between decision time and the action. So it's a very simple model, yet when we fit it to behavioral data, it kind of captures something in terms of behavioral curve and the chronometric curve. But as you see, for example, this outlier detection model is able has a worse prediction of the hit rate for weak changes than the mice do, which also kind of already implies that maybe mice actually use integration, because that will make it easier for them to detect those weaker changes. On top of it, we can use the stochastic nature of these fluctuations in temporal frequency to look how the combinations of two fast pulses with a certain interval between them influence the probability of these early leaks, right? And compare it to the independent effect of either of the pulses and see whether what we observe in the data matches the independent effect of pulses. So if it would match, that would mean that there is no interaction effect between pulses, right? Between the effect of pulses and the probability of a leak. And that would mean that mice don't integrate them. Yet we see that up to about 200 milliseconds between the pulses, there is definitely an effect that is unexplained by the independent effect of the pulses on the probability of the early leak. So all of this points out that mice integrate the stimuli at least on the scale of about 200 something milliseconds. Okay, so that's the behavior, right? Hope that's clear. All right, so now neural recordings. So me and Michael have performed recordings with inner pixel probes over majority of the brain regions. So what you see here is each point is a location of each good and stable unit mapped onto the Allen atlas of the mouse. Yeah, and so you can see, and here the heat map basically showing you the density of recordings. So it's not truly absolutely brain wide, but we covered the majority of the brain regions at least. And with that massive data set, how do you start analyzing that, right? So what we decided to do is to first to get an idea of what kind of behavioral variables are encoded where. We performed the generalized linear model analysis and originally done by Park and Pillow, also for decision-making task. And so what this analysis basically assumes is that firing rates of neurons represent linear combinations of behavioral variables, right? So for example, these example units from secondary motor cortex and trigeminal nucleus align to the onset of leaks, so just pre-stages of the activity. And in dashed line, this is the fit prediction of the GLM model, right? Just to show you that for most of units, the fit quality is very good. But the fitting, of course, is done on a single trial basis. And so the outputs of this model basically gives us this called kernels, which gives you kind of the temporal information of how different variables are encoded by the unit, if they are encoded. And to test the significance of whether a given predictor is contributing, right, to the variance of the firing rate of a given unit, you construct what we call the reduced model, right, which has all of the predictors except a target predictor, for example, leak preparation or leak execution. And we test whether we refit the model, and we test whether the remaining predictors are able to still recapitulate the overall PSTH, basically in this case. And if this fails, then this predictor has a significant contribution, right, to the unit's activity. So what do we see? Okay, so the top are just the maps of different brain regions, which are kind of color-coded according to the groups of brain regions. For example, the cerebellum in green, the basal ganglia in purple, and so on. And so here what you see is the, for each brain region, right, is a fraction of units that have a significant predictor of leak execution, right, or leak preparation. And so the fact that those kind of variables are very much close to brain-wide, at least in mouse, it's nothing new. Same was observed in original Steinman's paper on Anne Churchland lab. Now IBL, lots of papers show the same basically result. Also tangential but point for cerebellum, I'm lumping together a lot of things, right? So for example, if you see, I don't know, the simplex lobule or lobule 4-5, all of the units, right? I'm not distinguishing here whether these are Purkinje cells or Golgi cells or so on. I will have only one little bit of that towards the very end of the talk, but we of course can talk more about this. Okay, but so this is about leak execution preparation. But more interesting to us is how are these fluctuations in the stimulus speed are encoded in the brain, right? And so these maps show you exactly this. Notice that the top fraction is kind of lower, and overall the highest fraction of units are in early visual and high order visual areas, which makes sense, that's good. But what was surprising to see is that there is a sparse fraction, right? Say in frontal cortex, thalamus, hippocampus, cerebellum, but it's there. It's very, very broadly distributed. The only places where it actually is absent is some orofacial nuclei in medulla and olfactory areas, for example, and inferior colliculus. Right? So that was not very expected for us.
Can I just ask why, why wouldn't you expect that? I mean, the 20th century model is that regions of the brain separately encode things and there's not interactions, right? And
so, for example, in decision-making literature, often it's kind of implied that, for example, specialized cortical brain regions responsible for integration of visual evidence. We haven't got to integration yet, right? This is just representation of the kind of visual information. Still, that was somewhat surprising to us, but there is kind of a counterpoint to this. We're gonna get to this in a couple of slides, but basically one thing to keep in mind is that this is in trained mice that learn to utilize this visual stimulus. They know that they need to do something with it. So that's to keep in mind. Okay, so but to show you, this is just about whether the variable is encoded, right? But what are the temporal profiles of responses? And what we are talking about are basically the PSDHs, the average responses to the 50 millisecond long, either what we call fast pulse or slow pulse. So this is simply the instances in these fluctuations of temporal frequency that are higher or lower than a certain threshold relative to the mean. And to reiterate, this is during the period where mice just sample the stimulus and not making a lick. The earliest leak from this pulse is 2 seconds. That's done very intentionally. Okay, so 50 millisecond long event. In early visual areas, you see that those events also represented by kind of very briefly. But when you move to midbrain, high order visual areas, basal ganglia, frontal cortex, thalamus, cerebellum, you have a much broader response duration, right? And okay, and then just to prove to you that, you know, some people could think that, well, it's unexpected that it's such broadly distributed, so maybe there is still some efference of some tiny movements that mice make to those pulses of visual evidence. So if we look at from videography, if we look at the motion energy, for example, aligned to the mouse tongue touching the spout, we see a nice oscillatory kind of consumption of reward, yet we see nothing aligned to those pulses of visual evidence. Same if we look at the activity of units in the trigeminal motor nucleus, for example, right? Okay. So if we now characterize those responses to pulses of visual evidence along different brain regions, two main features that come to mind is the timing of the peak and the duration of the response, right? So if we look at the timing of the peak, unsurprisingly, it earliest occurs in the early visual areas and then propagates all over the brain. Similarly, the duration of the pulse of visual evidence is much shorter in early and high order visual areas than the rest of the brain, basically.
Andrei, is the superior colliculus here represented as midbrain?
So we split it. Yeah, it's a great question. So we split it. We put the, let me show you here. There we go. We put the superficial layers of SC into early visual because they get input from retina. But the middle layers and the deep layers, we put it to midbrain. All right, and to give you kind of visual representation of what's going on, so this will show you there we go, a 3D map. So every point is a unit, and if you see a point, it means its activity, the z-score of that unit is significant, and the size of the point scales with magnitude of the z-score, okay? And the color mapping is as usual. So visual areas are in blue and so on. Okay, so about like 200 milliseconds, visual already went through its peak and kind of starts to go down. Frontal cortex kicks in, cerebellum, sparse basic ganglia, midbrain. Now visual cortex is gone, cerebellum is on, frontal cortex is on, basic ganglia is on, deep midbrain, and now it kind of persists, right?
Andrei, for the cerebellar activity, is it just simple spikes, or does your model also include complex spikes?
So there are complex spikes, but I haven't seen any like what would look to me like a complex spike that will be TF responsive. What I can tell you though is that it looks like both what looks like Purkinje cells and non-Purkinje cells are TF responsive ones. Okay. But the question becomes whether this distributed nature of the stimulus representation, is it just because how the brain is inherently wired, right? So everything, if you trace a few synapses, is connected to almost everything. So no surprise. Or is it because that mouse brain has learned that stimulus has value? Or basically through learning, some modifications has happened to how it propagates. So to address this, we had some mice which were habituated to head fixation, but haven't been trained to do anything with the stimulus. So we showed them the same stimulus, but we just deliver them random rewards, right? Regardless of what they do. They actually, interesting enough, they their running diminishes through sessions. They just learn to observe, basically sit and watch the stimulus and don't need to do anything. So now when we do recordings on these naive mice, right, and apply the same GLM analysis and look what areas have those units that respond to pulses of visual evidence. This bar plot and this brain heat maps show you the same result, just in two different kind of versions. So here the solid bars show you the fraction of units active for trained mice, and the lighter bars show you in the untrained mice, right? So in the visual areas, we see actually a bit higher fraction in the untrained mice of units active. You can also see it here, right? And in some areas of the midbrain. But that's it. So interestingly, in frontal cortex, a little bit in striatum, but almost nothing. now in cerebellum and a bit in the hippocampus, right? So it does look like, so for example, the alternative to this was that this broad responses that we see, say, in frontal cortex, they would still be present, but they would be as narrow as they are in visual areas. But we don't see that. Instead, we see simply absence of response. So that was interesting. So that basically tells us that learning shapes the full propagation, and probably also the dynamics of response. We have some data towards that, but definitely the propagation of the visual evidence. Okay. So now we have this longer representation of pulse of sensory evidence in, say, secondary motor cortex. Why? What does it enable? Kind of obvious answer is that, well, this is well suited for integration of multiple pulses. And we have behavioral evidence that this is a strategy that mice use. So we can actually test it directly in data because we have randomly occurring instances of, for example, two fast pulses with a certain delay between them. And if we look at the responses in, say, superficial SC, as an example of early visual area, to a single pulse in black, and two pulses spread by 200 milliseconds in blue, right? You see that basically they're represented as two separate events. The height of these events is exactly the same because the response to first pulse is so sharp and narrow. But in the secondary motor cortex, because response to the first pulse has this long tail, the response to the follow-up pulse kind of adds up to this, right? And then we see this facilitation of response to the second pulse. And we can look at the magnitude of this facilitation as a function of delay between two pulses. And that's what we see, for example, for SC and for MOS. Right? So for SC, there is a little bit of facilitation if the pulse is sequential, but that's it. But for secondary motor cortex, it's up to like 400 milliseconds or so. There is something. And if we apply the same analysis across all of brain areas that have at least 10 of these TF responsive units, this is what we see. So what's shown here is this delta, right? So the facilitation of response to the second pulse relative to whatever is the size of response to the first one. So we see this facilitation, again, close to brain wide, but basically along the same brain areas that have long response to the first, to the single pulse of visual evidence. So midbrain, frontal cortex, basic ganglia, cerebellum, a little bit hippocampus. Interestingly, also in thalamus, it's VAL, which is part of the cerebellocortical lobe, and PF, which is kind of both cerebellocortical and basic ganglia cortical. So that matches nicely.
Andrei, is this a learned effect in the higher areas that is not present in the naive animals?
Well, so right. So yes, but it's I guess what I can say, right, is that in the higher order areas, is the response just not present, right? So it's not that it's narrow initially and then becomes wider. It just doesn't propagate there in naive animals. But it's very much plausible. We don't have a direct, we have something, right? But we don't have a direct proof that the way the task structure is likely shapes the integration times, right? So that's very, very likely, basically. What we can say is that it doesn't look like the integration times are determined by the intrinsic time constants. We looked into that. And I can show some figures if anyone would want to Okay. So this basically covers, so what we want is this kind of full transformation to cover, right? The sensory stimulus to integrated sensory evidence and interaction. And so far, we're doing kind of okay in time. We covered the sensory stimulus transformation into integrated sensory evidence, right? So the idea is that learning shapes the propagation of responses from early visual areas to almost the rest of the brain, where the response to single parts of evidence becomes much more prolonged, which enables integration of sensory evidence.
Andrei, I have a quick question, if you don't mind.
Yeah.
So the time constants you're showing here in the neural data seem quite a bit longer than what it is for the animal's behavior that you showed. So how do you interpret that?
Yeah, a good question. So for animals' behavior, I think it's about the amount of data that we have, because basically what we're working with are those instances of the two pulses. This is the main way we look at this. Two pulses that at some point lead to the early lick, right? But those instances, the number of n is extremely small. And so what we're able to pull out, if we go back to this behavioral plot, this thing, right? So this is not even from recording sessions. This is across the same mice as we used in recordings, but from training sessions. And this is over 100 sessions, right? And this is the kind of confidence intervals that we get. So I would say theoretically, if we have 100 times more data somehow, probably you would get to somewhat comparable timescales.
I mean, don't you have the reverse correlation analysis too on the so-called false alarms, which seemed like the SNR was much higher, which also is getting at the time constant, right? Yeah, this part.
You mean this?
Oh, I guess, yeah. Okay, so that's pretty long. All right.
Yeah.
Great,
here,
thanks.
one caveat is that part of this is just the decision to motor delays, right? But we have analysis also showing that it doesn't account for this whole width, at least.
Thanks.
Thank you.
Andrei, Farzana has a question. I wonder, Farzana, do you want to ask it, or do you want Andrei to read it?
I can ask it.
Please go
Yeah.
ahead.
Hi, Andrei.
Hi.
Good seeing you. Yeah, so I was curious if you go back to where you had the heat map with the facilitation. Right here. So I'm looking at some areas like DCN here or CA3. So these start with no facilitation, and then all of a sudden we see some facilitation. What's going on here?
Yeah, so here the map is thresholded by the significance of effect, right? So basically, you see the confidence intervals. So roughly what this is that this confidence interval needs to be outside of the 95% higher confidence interval of the single pulse, right? So we have a significant facilitation, basically. That's what it is. So for DCN, it's just number of n, right? So we had the threshold to include into this figure of 10 TF responsive units. And this is exactly how many we had in DCN. So it's just bad luck. I think personally, this is just low number of n, right? So it's basically uninterpretable, I would say.
Okay.
Yeah.
Thanks.
Yeah. Okay. So now going into the sensory evidence introduction. First, I want to give you the visual idea of what's going on. So we're looking now at the kind of same type of the activity. Each point is a unit that's showing the magnitude of the Z-score. If it's significant, we start at 1.2 seconds before the lick, and then we progress towards the lick. Right? So much less sparse than the single little pulse of sensory evidence was. Frontal cortex, basic ganglia, thalamus, midbrain, higher order visual, even basic ganglia, of course. And now everything kicks in, right? So this efference of action, like everyone's showing, it's going basically everywhere. Okay, so how do you address this, right? So our thought process was, well, if we want to look at the transformation into action, we might as well use the ideas from the motor control, population analysis ideas. And so here we use the same framework as done originally by Kaufman, Churchland, Shenoy, and Rue, where they wanted to map the activity from the premotor cortex and primary motor cortex neurons into the deltoid or the EMG activity of muscles during the center outreach task. So their puzzle was that the activity of deltoid EMGs is basically 0 before the go cue, and then there is some activity, yet there is the strong preparatory activity in the, say, frontal motor cortex. And so they reconcile this by introducing this movement potent and movement null mapping. So this basically is just a linear algebra, right? Where you have some main modes of activity. So this is low dimensional activity M of the EMGs. And so what this does, it basically, you can think about it as a linear combination of the activities of your target brain area that adds up with certain weights into this profile of activity that the EMG has. Okay. The cool thing is that dimensionality of neural data is, most cases, more than the motor output. And so what this means is that there are orthogonal dimensions, orthogonal linear combinations of the activities of units. That's what's called the movement null subspace, right? The projection of those combinations into this movement potent dimensions is 0. And so what they saw is that when you project the activity onto this movement null dimensions, this is where the preparatory activity, population preparatory activity, resides, right? So this was the answer, basically. How does this preparatory activity not trigger immediate reach? It doesn't trigger because it adds up to 0 on this movement potent dimensions, but it's very much non-zero on this movement null dimensions. So I wanted to apply the same framework to our data. And as a side note, we are talking now about leak aligned activity and its leak onset time from the videography, right? So you can see here motion energy as a function of time. And so this red line is where our estimated onset of any basically orofacial movement, to be fair. Not necessarily like just jaw movement. So that's what we call the leak onset time. Okay. Now, if so, I don't have EMG signals, but I have activity of what we call the orofacial nuclei, and here it's a bit the combination of trigeminal, IRN, and then SPVI, SPVO. So it's a bit of a mix, right? Because ideally we would have wanted like trigeminal and hypoglossal nuclei. We don't have recordings any from hypoglossal, but also so these are more like sensory nuclei, right? If I remember right. But overall, what we want is some nuclei that just represent the movement, right? Whether they drive this orofacial movement or get the direct sensory efference of it. That's okay for us. We just want the overall population activity there. Okay, so if we look at the projection on top principal components of, for example, this orofacial nuclei and the secondary motor cortex, what we see for orofacial nuclei is that they don't have any preparatory activity, at least not to the extent that secondary motor cortex does. And it's overall low dimensional, right? So you see that basically the orofacial nucleus is basically close to one-dimensional, but we use 2 dimensions here just to make it more interesting. And so what this means is that if this movement potent mapping, right, defines the mapping between the 4 principal components, the projections on 4 principal components for secondary motor cortex, such that they add up to the profile of projection on this, or same as orofacial nuclear activity. So here you see the result of this mapping, right? So this defines what we call the movement potent subspace. And also to note, the mapping is done from basically only during this expression of the response, so from the up to like 100 milliseconds before the leak onset. So in principle, if the preparatory activity has a chance to kind of still be localized to the movement subspace, yet we don't see this. Now, if we look at the 2 remaining dimensions, which define the movement null subspace, we see for now secondary motor cortex, same result as Kaufman and Churchland saw, right? So this is the preparatory activity is in the movement null subspace. And the only trick I do here is I make a rotation within this movement null such that the preparatory activity is maximized on the first dimension. Okay, so now when we apply the same analysis for each brain area, right, separately. Okay, so this is what we see. So what's shown here for every area is a peak normalized Euclidean distance in the movement subspace and movement null subspace from these 2 seconds before the leak onset, right? The way you read this is basically if it's white, then there is not much going on at this time point, right? So for the movement subspace, you see that there is almost no spillover of any preparatory activity in these 2 dimensions. And it's all kind of localized to this movement null subspace, right? And that's not forced in any way. And moreover, if we look at basically the kind of ratio between that, so the relative occupancy, whether the activity is more within the movement null or movement potent subspace at every moment of time, we see that if there is a preferential occupancy of preparatory activity, it's always in the movement null subspace. So we kind of confirm and recapitulate the original finding of Kaufman and Shadmehr in Churchland, but by for essentially all of the brain areas that have preparatory activity. So that was somewhat unexpected, but nice. Okay. Now the final bit is we want to we're still studying decision making, right? So we want to understand how sensory evidence drives or relates to this preparatory activity. So for that, we decompose the projections of any of these dimensions into sum of 2 contributions, right? So because basically what defines a dimension is the set of loadings, the weights, right? And we know which weight corresponds to which unit and whether that unit is responsive to sensory evidence from the GLM analysis or not, right? So this is how this decomposition of projection is done. And black is projection of the full population, red is TF non-responsive units, and cyan is TF responsive units, right? So what you notice here is that though there is only about 5% of TF responsive units, they contribute in terms of this preparatory activity about as much as the rest of the population. And the whole analysis is done on the full population, right? So that's again not forced in any way. And this is the same, and this is a projection just on the other second movement-potent, movement-null dimension to show you that this is where the TF responsive units basically main activity lies. Okay, and this is the same thing in the state space, right? Just to visualize that if you look at the first movement-potent and movement-null dimensions, all activity starts here. The TF responsive units, they basically propagate only along this movement-null dimension, right? There is almost nothing here. And okay, and to quantify this, we compare the contribution of the evidence responsive units to the random sample of the same fraction of units from the whole population, right? So that basically compares to whether what they contribute is more than you expect kind of from uniform coding of the whole population. And that's what the gray line shows, right? And so this green line here effectively shows kind of a ratio, but this is single dimension. We work along 2 dimensions, the whole subspace, but this is what roughly it is. How much the TF responsive units contribute of the full population vector, right? So it's roughly the ratio of that. And gray is again shows you the chance level as before. So what you see here is the peak, kind of the quantified version. There's the peak of the contribution happens right around the onset of the leak. And then interestingly, it sharply collapses. And we can repeat the same analysis for all brain areas. And so this is what we see. So the color shows you the relative contribution of TF responsive units. And the color coding is the red is basically the same alignment as the main population vector, the full population vector. And the blue is some cases where TF responsive units point in the opposite direction, which is not what you expect, but it's possible. That's what's happening. Now, so what you notice is now this set of areas that have over chance contribution of TF responsive units is much more restricted than what you had before. But in terms of what kind of areas, it's still cerebellum, basal ganglia, frontal cortex is still a mix, right? It's not a single brain region. Even more interestingly, if you look at the time profile of the contribution, it drops to basically this chance level about 100 or so milliseconds for most areas after the leak onset, despite this presence of the sensory stimulus, right, of this increase in the stimulus speed. So that means that this increased contribution, it's not just because the sensory stimulus is present, it's because it's relevant to the ongoing computation, which is this upcoming action. And moreover, if we project the population responses to pulses of sensory evidence to either single or to sequential, fast or slow pulses to this movement null or movement potent dimensions, we see that as expected with preparatory activity, the pulse size and sign scale along this first movement null dimension, exactly where the preparatory activity lies. And we see the same alignment between the fast pulse and the movement potent, movement null, but not movement potent dimensions for basically all brain areas.
Andrei, I had a question. One thing that's sort of puzzling to me is the licking is a rhythmic behavior, and I don't know if I saw in the movement potent direction any rhythmic activity associated with your.
So So that's interesting. It is rhythmic if we align it to there we go. Scroll a lot. There we go. To the time when tongue touches the spout, then it's rhythmic. But if we align it to the onset of the lick, right? So the first instance when we see some from videography, some facial motion, then it actually, like, this is rhythmic even on average, right? But if we do the alignment to the onset of the licks, it's no longer rhythmic. I think it just smears all this rhythmicity. Yeah, I don't have single unit examples, but this is the population, the projections and the principal components.
So Andrei, just to clarify that, so is the idea that what you want to measure is the time that he decides to do his licking bout?
Exactly.
And okay.
Or the first hour kind of identification, right? The decision has been made that the action starts. Yeah. So the overall idea is that the integration of these pulses along this first moment null dimension is what creates this activity of TF responsive units. And then through some internal dynamics, they drive the rest of the population. And that's how this ramp progresses. Okay. And as a final bonus, so this is some very rough analysis, but still. So this is what happens if I split, for example, recordings in lobule simplex into units that have high baseline Fahrenheit rate, so very putatively we call them putative Purkinje cells, and the rest, right? And apply the same population level analysis. You see that this ramps along the movement now kind of much longer on the pre-Purkinje cells than Purkinje cells, which is exactly what you would expect. So it's not surprising, but it's kind of consistent as what you expect. But of course, the proper way to look at this is with complex spec identification and on a single session. All right, so with that, I just want to thank all the people that contributed to this project, especially Michael, who is a joint author, and everyone else who helped with either training or modeling, and Tom, of course, for all the mentoring and support. Thank you.
Thank you, Andrei. That was fantastic. Thank you for the slow pace with which you walked us through all that data. And let's see if we had lots of questions for you already, but if you have any questions, just go ahead and raise your hand and I will call on you, or anything that you want to ask Andrei. Let's go ahead and do so.
Hey, Andrei, really amazing data. So my question's about individual variability. Presumably some mice sort of integrate more information, some less to make their decision. And I wonder if you see any signatures of that in the timescales of the neurons across the brain.
That's a good question. So just to give other people context. So for example, the International Brain Laboratory, they did similar analysis, and there are very strong different biases, right, of different mice. In our case, there is less of an option of bias because it's a go, no go task. Let's go here. So the short answer is not really. So for example, this leak triggered average of the licks, I don't have, unfortunately, the bonus slide for this, but they're extremely similar across different mice. The only caveat, I guess, is that these are all highly trained mice that kind of progress through all the stages of training and pass all our check marks. We don't explicitly force the integration, but they need to detect with this, with a certain like fraction correct the higher changes to progress further in training, for example. So perhaps mice that, for example, would just kind of ignore the sensory stimulus and just use the estimation of average timing of this change period would just score less, and they would never progress for me to do recordings in the end. Yeah.
Andrei, Farzana asked that if you could go to your summary slide so we can look at that. And Sasha, go ahead, please. You have a question.
Sure. Thank you, Andrei. Of course, I'm terribly disappointed about very few cerebellar nucleus neurons, and the nuclei are really different. So you wouldn't expect the same thing. I wouldn't lump them. But this is just the mildly critical prelude to the question, which is, well, where do you go with this? You know, like, what's the question? So now the answer is something like decision making is distributed, and during the preparation doesn't contribute to the movement per se. But like, you're a smart guy, and I really want to know where you are going to go with this. I don't mean to criticize the entire field to you, right? But like, help me make a decision, you know?
Well, so part of it, I guess, it creates an idea, right? So, for example, of what. So, okay, one thing that is hard to disentangle from any this type of analysis is whether the region is necessary for the implementation of all these computations, or whether it just gets an inference, right, of this information. So this type of analysis, though, for example, this sort of latency for a reason, right? So these earliest regions, they are likely to be causal, for example. So this gives us some kind of early candidates, right, where to look for either this integration of the sensory evidence or for this switch in the modes of activity. So, right, because this like we can think about this preparatory activity as overall accumulation of sensory evidence, fine, but then how does it exactly trigger the actual action, right? So in the motor control, the idea is that, well, motor context is just a dynamical system. You just provided the initial state and then somehow it just does the thing. But studies that actually silence the input from thalamus to cortex, they don't really support that, right? So the direct answer is that you can start addressing this now only with perturbations, right? With either optogenetic perturbations to see the deficits in the dynamics of the downstream regions or the deficits in behavior.
Okay. So, and
it's okay. Let me go to the next question, Sasha, if you don't mind.
Of course.
Jennifer, please go ahead.
Hi, Andrei. Can you hear me?
Yeah. Hi, Jennifer.
Nice to see you. Yeah, really interesting finding about the change in time constant for integration. And I think this is related to Sasha's question or your answer to it, but are you expecting that in each brain area where you see this increased time constant, there's something happening in the way that area is integrating information, or are you thinking there's one area that does that and then passes the signal to everyone?
So I think it's very likely that it's very unlikely that there is a singular area and then everywhere else is just an inference, right? It's likely to be, at least to a degree, a distributed computation. And so the clue for this is all of our versions of plots, and not only ours, like or same from, say, Brody Lab, that look at the latency of any sort of computation distributed, right? We see something like this. So like, for example, cerebellum, basal ganglia, frontal cortex, and some thalamus. So I think this is this long-range loops that actually do the job. And then it's, I guess, back to Sasha's question, the puzzle would be to disentangle, right? How the different modules in this either basal ganglia, cortical, or cerebellar cortical loops actually contribute to this different parts of the computation.
Can you test this with stimulation? If each area is increasing its integration time constant, then can you activate excitatory inputs to a given area and look for that directly?
Kinda. I mean, I think okay, let me show you one thing. So what we did look for, so there we go. This is kind of the same question, but we're trying to look at the different angles. So here the question was whether the what's called the intrinsic time constants in the brain regions determine the duration of the integration of the individual pulses. And so intrinsic time constants, they're simply the time constants of the autocorrelation of activity that happens during the late parts of the intertrial interval, when mouse just sits still in the gray screen and sees a green screen. And there is a literature that kind of goes along this line, that's the intrinsic time constants determine the overall time constants of dynamics, right, during some presentation of the stimulus. But we don't see that. What we do see, though, is if we estimate time constants, intrinsic time constants, for both trained mice and naive mice, they match very well. So they're indeed intrinsic time constants. So what this points, that's why I was speculating, right, that the probably exact way, the exact temporal statistical structure of the task very likely determines the time constants of integration, which would make perfect sense, right? Yeah.
So mechanistically, what are the, what are the range of possibilities? Some rapid change in intrinsic excitability, some change in the time constant of an extrinsic synaptic input.
I mean, if just
what are the things you're thinking?
Yeah, just speculating, but in principle, it could be, say, it could be in partially cerebellar, right? Because the time scales are cerebellar. And so if the idea is to kind of amplify the cortical loop for a certain amount of time, this is exactly what cerebellum can learn. But of course, it's not, all of this analysis, I'm lumping a lot of things together. This is very unlikely to be like a global computation. It's much more plausible, I think, that there are some specific modules on the cerebellum that were specialized for that.
Gabe, let's go to you for our final question.
Okay, sorry for asking two questions, but I'll try to hopefully be clear about this. But if I understand the task structure correctly, I believe it's the case that the animals, as time goes on in the trial, the threshold for committing to a decision should decrease as time goes on. Just if you think about a hazard rate, right? Like the longer time goes on, the more likely the change is going to occur at this time, given that it hasn't occurred yet. Mm-hmm. And so what I'm wondering is if you specifically look at trials where the change occurred late and the animal did not respond early, would you see that the this alignment between the evidence direction in state space and the movement to null direction would be less well aligned because later longer trials it would be more time is contributing to decision rather than the pulses themselves? I hope that makes sense.
It does. I think it's kind of a variant of your question is that if I, for example, split trials by the reaction times, right? So, and some, well, this is Lick aligned, but if you look during the change, there are some hits that happen earlier, some hits that happen later. So why is those that happen later happen?
Well, just to clarify, the reaction time is time from the change to the lick, right?
Yes.
So what I'm talking about is time from the change to the beginning of the trial.
Okay, so you're talking about right.
Right. So the mouse has a hazard rate. Like if nothing has happened, like given at this point, like as time goes on, it becomes more and more likely that something's going to change.
Okay. Yeah. So this is something that we kind of ignored for this paper. We're looking and on the small next one. Sorry, lots of scrolling, but I just need to show this. So there are actually two blocks of when the change happens on our task, what we call the early block and the late block. And as you say, exactly, mice learn the temporal expectation, and the temporal expectation has a different hazard rate. So if we look at the timing of the early licks, right, the hazard rate is very different on these two blocks. Now, the projections of, so I've done that. The projection of early licks in either of those cases on the movement potent movement null dimensions defined by the hit licks matches very well. I have it not split by blocks, but I should have one bonus slide. Oh, there we go. Okay. Yes, threaten, why not? So this is not split by early and late, but just kind of trust me that it doesn't change drastically. So this is projection in green on early licks, right? On these two dimensions. So sometimes they're not exactly reach the same magnitude of the peak, but qualitatively they're very similar. Now, what does change in terms of the early and late blocks, in terms of hazard rate, is we see those ramps that start from the trial onset. And in large, and the slope of those ramps is different on those blocks. So and that basically sets the initial condition, right, of the licks differently as time goes on the trial. So we think this is roughly the neural implementation of that.
All right, thanks. I'll have to think about it more, but appreciate it.
Thank you, Gabe, and thank you, Andrei. That was really just a lovely presentation, and I'm so grateful for you walked us through the results and beautiful work that you've done. Thank you, everyone. Have a wonderful day.